I'm Kelly Salmon. I'm the senior scientist for the New Hampshire Academy of Science. We are a nonprofit. We operate a lab in Lyme, New Hampshire, where we allow middle and high school students to go through the whole scientific process. So they have ownership over the lab, they come in, they work on their projects. But we've pivoted this summer to instead having them work on their projects at home, and we're loaning out equipment and mentoring them over Zoom and things like that. So that's a little bit about what we do, and we do outreach like this. So this is continuing our series on COVID-19, and tonight we're going to be talking about vaccines and the race for a SARS-CoV-2 vaccine. And just to give you a summary before we get started, we're going to talk a little bit about the history of vaccines, then dive into what exactly are vaccines and how do they work, how do they get tested and approved, and then about the SARS-CoV-2 vaccines that are currently in development, which includes a couple of types that we've never actually had vaccines on the market like them before. So first, some vaccine history. It really goes back to smallpox. So this was a really terrible epidemic that was going on for thousands of years. It left 2060, or killed 20-60% of people who were infected, which turned out to be about hundreds of thousands per year. It left survivors scarred, some were blinded, but people noticed that people who did get smallpox didn't get it again. And this led to an idea of basically purposely giving yourself a small amount of smallpox. So this was first done through variolation, where it was this controlled exposure to the pustule or the smallpox from another individual who was infected, either a dried pox that was blown up the nose, gross, or some pus that was taken and put under the skin of the individual undergoing variolation, which is somehow less gross, but both are very gross.
Yeah, Cameras is always making funny faces when he hears about this gross biology stuff.
Yeah, there's lots.
So gross.
Lots of gross biology. And this was actually practiced for quite a while in Africa and Asia before being brought west in the 1700s. And Lady Mary Wortley Montagu is best known for bringing this back from Turkey to Britain. And there was also a slave who is only recorded as Onesimus, who introduced it to Massachusetts during a smallpox outbreak there, also in the early 1700s. And this was still really risky. So about 2% of people who underwent the procedure died. You could still fully contract smallpox. You could still pass smallpox to others. And on the left, or on the right here, on the left of the right image, is showing a variolation. So you can see the person still getting some pretty nasty smallpox infection, though theoretically less severe than they would have by contracting smallpox regularly. And this is compared on the right to a proper vaccination, which we'll get to in a second, which still, we're talking 1800s here. It's not the prettiest of vaccinations, but a whole lot better than having a smallpox outbreak on your arm. And another smallpox observation that a lot of people were making at the time was that milkmaids who were exposed to cowpox from cows rarely got smallpox. And cowpox is a vaccinia virus, so you might get where we're going here. This is going towards a vaccine. And though he was not the first person to make this connection between the milkmaids not getting smallpox and the cowpox, and probably not even the first to try inoculating someone with cowpox to prevent smallpox, he actually designed an experiment, carried it out, and published the results. And that's Edward Jenner, who's shown here. And in 1796, he took pus, again with the pus, from a milkmaid named Sarah Nelm, who had gotten it from a cow named Blossom. I'm very glad that this has been recorded. And he put that under the skin on the arm of an 8-year-old boy, James Phipps, who is pictured here. We'll talk about clinical trials and stuff in a little bit. This would definitely not be approved now to pick an 8-year-old boy as your target and have one person as your experiment. But this was the son of his gardener, and somehow this kid got roped in. So he had cowpox put under his skin in May, and in July, Jenner purposely exposed the kid to smallpox, and found that the kid did not get infected with the smallpox. He did also note that after the inoculation with cowpox, the boy did have some reaction. So he had swollen lymph nodes, he had low-grade fever. So all this was really well documented, and he published it a few years later. And this led to a flurry of people doing this across Europe, though even then it was controversial at the time as to whether people should be doing it and should be forced to undergo vaccination. But in the end, better vaccinations than what Edward Jenner had done eventually eradicated smallpox. So this is showing you the last time that a case of smallpox was diagnosed on every continent. And the last case was in 1977 in Africa. So because of large-scale vaccination worldwide, we've actually completely eliminated smallpox from the human population. And with vaccines, we've nearly eradicated polio worldwide, which is probably actually going to be set back by the current COVID-19 pandemic with people not getting their vaccinations. And in the US, it has largely decreased the amount of cases of diphtheria, measles, mumps, rubella, though these do crop back up, especially in small pockets where people are not getting their vaccinations. So what exactly are vaccines and how do they work? We'll go into more detail about all these steps, but just as an overview, you are typically injecting a weakened or dead version of, this says bacteria, we're actually talking about a virus here and it can be either, into a person. And this is often done by injection if you think about the shots that you've had before or the vaccinations you've had before, but there are some oral ones or nasal ones. And this weakened or dead form is able to trigger an immune response and produce antibodies. So we talked a lot about antibodies last week, so I'm going to go through a quick reminder. But these are a large part of your immune memory and allow your immune system to remember things that it's encountered in the past. And this immune response that is generated against this weak or dead version of the pathogen is able to stick around in the patient. And then if the patient encounters the actual disease later, they're primed to respond to it well. And I showed this last week as well. It's just showing you here antibodies made by an individual over time. And so this is looking at your adaptive immune system, which produces antibodies as one of its ways of battling off invaders. And the first time you have a virus attack you or you get a vaccine, it takes you a little bit of time for your immune system to respond. And then you ramp up a response and it goes back down again, but lingers. So you still have some antibodies present. And then if you get attacked a second time by that virus or the first time after vaccination, you have a much quicker and much stronger response. So this could potentially keep you from getting sick with the virus. And as a little bit more of a reminder about what antibodies are doing, they are often neutralizing, and this is a lot of what you hear about when people are talking about the clinical trials right now. They often mention neutralizing antibodies. And these are just binding to, we're going to focus on the virus bit over here, in a way that prevents it from doing its function. So if we think of this little virus cartoon as SARS-CoV-2, these spike proteins coming out are really critical for it binding to the human cells and getting entry. And if those sites are occupied by these antibodies, then it can't do that as well. And the antibodies also help signal to the immune system to clear this thing that they're stuck to. And this can be through something called opsonization, where you have other immune cells come and gobble up that thing, or through other parts of your immune system, such as complement, that can destroy the invader. It can also be cleared from your blood. So what do we want when we're making a vaccine? We want something that actually protects you against the disease. So in our case, we want to defeat SARS-CoV-2 fast enough that we're not getting COVID-19. And preferentially protect against multiple forms. So if there are different forms of a virus or bacteria or anything out there, or if it's something that can mutate, you ideally want to protect against all those forms. You also want long-lasting immunity. And we see this vary in different vaccines that we get typically, where some you need to catch up on. So like tetanus, you need to get every 5 to 10 years, whereas other ones you can get as a kid and you're basically good to go. But ideally we want the longer lasting. We also want it to be easy to administer so people will actually take it. And you want to work on all age groups, ideally, and most individuals in general. There are always going to be some people in the population who are immune compromised or have specific allergies, things like that, that they will not be able to take a vaccine, but you want the majority of the population to be safe taking it. So this is a busy slide. We're going to go through it slowly. There are a couple different types of vaccines that are currently in use, and we're going to talk about the newfangled ones in a little bit. One is inactivated virus or killed virus. And I put the killed in quotes because, as we talked about, I think 2 weeks ago, viruses are not quite alive. So they can't really be alive or dead. They need to take over a host cell to be able to replicate. But the killed here just indicates that it's not able to do that function. It's not able to go in and replicate in a cell and take over. And this really gives you the most durable type of immunity because you're basically giving the whole virus to the immune system to figure out how to learn how to fight it. And it is the best trigger for the immune system because it looks like a real virus. These do unfortunately often require cold storage, so they're a little bit harder to get to more rural areas and administer to more remote populations. And some examples of ones that people are familiar with that are in this category are the MMR vaccine and chickenpox vaccine. Another type is attenuated or live. So this is not a killed virus. It is still able to do its function, but attenuated means that it's been really heavily weakened, and usually weakened in a way that it is not able to take over a cell. These are actually a little bit less immunogenic, so less prompting an immune response than the inactivated virus. So it's a little bit more likely that you'll need a booster shot to make sure your immune system kind of gets the point, or adjuvants, which we'll talk about more in a second. But it's still a really good immune boost to make your immune system learn what it's trying to fight. And some examples that people might be familiar with from this category are the polio shot, not the oral vaccine, some flu shots, there's quite a few out there, and the vaccine for Hep A. And I'm going to reference my little diagram on the right. So we have our pathogen here that would cause disease. And the replicating live attenuated pathogen, so it's live because it can still replicate, is very good at prompting immunogenicity, so prompting an immune response, but it can be a little bit less tolerated. So these are inversely correlated, the amount of immune response you get and how well tolerated it is by the body. So you might have more of a negative response to something like a live attenuated or a non-replicating inactivated virus, but you get a much better immune response. However, this means that if you are someone who already has a struggling immune system, you might not want a vaccine like this. And there is a small chance with any of these live attenuated pathogens that they could revert. That I think we've gotten pretty good at making sure that that does not happen. The third type is subunit or antigen. So we talked about antigens last week. That's just a thing that your immune system can pick up on, so a part of a virus. And subunit would be a chunk of a virus, so kind of chopped up virus chunks floating around. And these are also prompt less of an immune response because they're not the whole virus. It doesn't seem to stimulate your immune system in the same way. So these are more likely to need boosters or adjuvants to make sure that the immune system gets a sufficient response to remember that, that antigen. And some examples of these are the Hep B vaccine, hepatitis B, the HPV vaccine, and the shingles vaccine. And this is showing here that basically as you get to smaller and smaller bits of the virus, so going down from subunit to purified antigens, you're again losing some immunogenicity, but you're gaining some tolerability. So a purified antigen vaccine tends to cause less aggravation to the immune system.
Kelly,
Yes,
can you explain what an adjuvant is?
yes, I think it's the next slide.
Oh, sorry.
It's okay, yes. So adjuvants are things that you're putting into the vaccine along with your purified antigen or your virus that are basically there to aggravate the immune system and to prompt a better response, so that hopefully the immune system will better remember that little, instead of just being a little protein that the immune system maybe responds to and is like, that wasn't a big deal. This adjuvant is causing enough other things, enough inflammation usually, that the immune system is able to better pick up and remember the antigen that is along with the adjuvant. And this can be really good for, and this is going to become important possibly with the coronavirus vaccines, is that you can sneak less of your target into each vaccine. So if you're thinking about making a couple billion doses of a vaccine, including something like adjuvants may become important. But it's just a better way of basically taking this purified antigen and making it a little bit better at prompting an immune response, because you're giving the immune system something else to react to as well.
So I'll chime in here if you don't
Yeah.
mind. These when I made animals immune to things by giving them vaccines that we made ourselves in my old company, we often had to give adjuvants because they were terrible antigens. And the most common adjuvant today now is actually an aluminum salt. So aluminum hydroxide, I think, or aluminum phosphates, and we don't really understand how they work. But they're the minority of the 40 or so vaccines that are in the United States now use adjuvants. Most of them are actually pretty good on their own, but some of them don't work very well. Some of the vaccines don't work very well, so we do have adjuvants. And most of them, I think, are aluminum based, and they're pretty benign.
Yeah, most are aluminum, and it's been used since the 30s, I think. And there's not been any reported negative effects. There are a few other adjuvants I came across, but they're really either were used in like one vaccine and are no longer used, or are just kind of an option that aren't really in a lot of vaccines on the market. So this is kind of what Peter was getting at. So this is showing you the vaccines that are licensed in the US, and ones that have adjuvants are in blue, and the ones that don't have adjuvants are in red. So a lot of the older vaccines, even though adjuvants were discovered somewhere, this is showing somewhere in the 1920s. Oh yeah, the aluminum was the 30s. The adjuvants were discovered in the 1920s. A lot of them don't have adjuvants, but more recently we do have some, and that's probably also because we've gotten better at purifying the individual protein bits to include in vaccines, rather than having to grow up a lot of virus and heat kill it to make an inactivated form, or do something to attenuate it over time.
I think also, if you look at in between 1940 and 1980, we hit all the real major, these are the really major diseases that needed vaccines. And Economics 101, you knock off the easy ones first.
Yeah.
and then you get into the tough ones.
Yeah. Yeah, and that's kind of where we've been. I mean, we've been trying to make an HIV vaccine for decades, I think now, and that's a really tough one.
Yeah.
Okay, as if all this wasn't complicated enough, there have been instances of vaccines that overactivate the immune system, which can actually make the disease worse if an individual actually comes in contact with the virus or the bacteria. And this is called immune enhancement. It can happen through a few different pathways. And unfortunately, there was a bad example in the 1960s with RSV, or respiratory syncytial virus, where children received a vaccine for RSV, and when they encountered it out in the world, a little bit later, a lot of the kids got really sick. 2 kids died. And it really was a wake-up call for looking for those types of reactions to vaccines. So this is just something else that people need to look for when we're looking for side effects before doing a wide-scale release of vaccines. And this, I have seen this referenced a few times in talking about vaccines for SARS-CoV-2. So that's explaining this. So far, there's no indication that SARS-CoV-2 infects immune cells in a way that immune enhancement is likely to be a problem, but we just don't really know yet. And a last thing about vaccines in general before we jump to one specific to SARS-CoV-2 is pointing out that vaccines are important for the individual to keep the individual from getting the disease, but it's also really important for a population as a whole. And that's because if you have a lot of people, so if we start over here with no immunization, we have a lot of people, one person, or actually 2 people, are not vaccinated and are contagious with a disease. You have a lot of healthy non-vaccinated people and a very few healthy vaccinated people or people who had the disease and recovered. And in this case, the virus can spread really easily. If you have immunization and a lot of people have already, their immune systems have seen the virus and know how to fight it off, you can still have a few people who are not immunized, these blue heads over here, but they're still largely protected from a person who is infected. And we can look at this from a math perspective, and this gets referenced a lot in the news as well, the R0 value, or R0. And all that is the average number of people that a single person with the disease infects. So this can range, and they're still trying to figure out what the proper number is for SARS-CoV-2. But for example, mumps here is about 4 to 7 people per person infected. And measles is crazy contagious. A single person can infect 12 to 18 people on average. And this the R0 changes the threshold of people in the community that need to be vaccinated in order to protect the community as a whole. So the more contagious a disease is, such as measles, the more people you need to get with a vaccine in order to protect the community as a whole.
Yeah, and that is a great point, Kelly, and it can't be overemphasized because we do not really know what the R0 is for COVID. And because of that, we do not know what the herd immunity requirement in percentage is going to be. Yeah. We're really in the dark here.
Yeah.
It took a long time for us to figure out these other diseases have been around for hundreds, if not thousands of years. So it was easier for us to get an R0 for these. And measles and pertussis, we know, as you know, whooping cough pertussis is transmitted by coughing. Measles, same thing. Polio, same things. These really contagious diseases are spread by, coughing. It's aerosolization of the virus. So any of these things that are transmitted in the air are really a problem, and that's why COVID is such a tricky thing to deal with, and we just don't know what the R0 is.
Yeah, and as I mentioned with vaccines, also not everyone can get any given vaccine. So say you're undergoing cancer treatment, you have a weakened immune system, your body can't, even if you got a vaccine, you couldn't get a good immune response. So there are people who just can't get vaccines and be protected that way. So they rely on the population to get that protection. So that is my last big point on vaccines in general. And then we'll switch specifically to talking about the ones for COVID-19. And just as a reminder of our target, so this is our SARS-CoV-2 virus, and it's got RNA on the inside that encodes for everything important for the virus to function. Then it's got a membrane, and the main thing here is these spike proteins, because as the sticky outy bits, that's what your immune system is likely to pick up on and recognize. So that's really what we're mostly trying to target an immune response towards. And this is also the protein that's important for the SARS-CoV-2 virus to bind to human cells and get in. So we also want to block that function. And as a lot of you or all of you are probably aware, there are a lot of horses currently in the race for a coronavirus vaccine. And the New York Times is actually running this pretty cool coronavirus vaccine tracker that gets updated really frequently. This was updated yesterday when I was working on this presentation. There are over 160 vaccines that are currently in development. Most of these are still preclinical, and I'll talk about the different phases of trials in just a second. But we already have vaccines that are in phase 1, phase 2, phase 3, and one approved for limited use. This is in China, specifically for the Chinese military. So to talk a little bit about clinical trials, this is our way of testing drugs and vaccines before they get introduced to the general population. And it's a way of doing human subject testing that we can figure out if a drug or vaccine is safe and if it's actually effective. And we do this in phases so that we minimize the number of people exposed until we learn more about the drug or the vaccine. So preclinical means that the tests are still being done on animals, lab animals. Then once you get past preclinical and show that it's safe in laboratory animals and seems to have a, we'll talk about vaccines specifically, so seems to have an immune response in animals, then you can go for a new drug application through the federal government and go into phase 1 clinical trials. In phase 1, you're typically in the tens of patients, so 20 to 80 patients. And this is largely for safety. So you're typically testing out different doses of your vaccine or drug and seeing, do people have an adverse reaction to that drug or vaccine? And in our case, we can also look at these people for, are they having an immune response that we would expect from a vaccine to SARS-CoV-2? Once a drug gets past this initial safety test, it moves on to phase 2, where you're just increasing the number of people that are being exposed to a couple, dozens to hundreds. And this is to get you a more broad population to look at for adverse effects. And you start looking more at efficacy. Are we actually getting the desired outcome? Phase 3, it goes into the hundreds to thousands of patients. And we'll talk in a second about a few drugs that are about to go into phase 3, and it's going to be about 30,000 people per trial. And this is a lot for efficacy. So you're going to have a control group that gets a placebo, basically a fake injection if we're talking about something that's injected, and a group that gets the actual vaccine, and then see how many of each group become infected. And look at their antibody response. Are they producing antibodies like we would expect if their immune system is remembering this virus? And if you get through all 3 phases, then it goes into a review and approval process. For the SARS-CoV-2 vaccine, this is this is all being accelerated, as we'll talk about in a second. And so will the application review and the approval likely. And typically, a lot of drugs and vaccines get weeded out. This is looking specifically at drug trials because there's a lot more of those typically than vaccine trials, get weeded out during these clinical trials. So we lose a bunch, about a third in phase 1 trials, just it had adverse effects that were too much for whatever the drug is supposed to do. We lose even more in phase 2 once you actually start looking at efficacy and does it do what we need it to do. And you lose some more in phase 3. And at the end, you're getting less than 10% of the ones that you initially started with that actually make it to market. And as I mentioned, everything right now is very condensed. So I'm calling this pandemic speed. Typically, the vaccine development process takes about 10 years. The fastest we've ever done this is 4 years. And we're doing that by taking all these little blue bars, they're typically one after another, and pushing them under each other so that we have multiple things going on at the same time. So typically there's a lot of research about whatever the disease is before you even go into this vaccine. And we just discovered this disease. So we are moving very quickly into vaccine development right after discovery. So there's a lot of academic research going on right now, but we started moving well into the vaccine world before that research got very far. Then in those clinical trials, we're actually combining phases. So I'm going to talk about in a little bit, and I'm sure you've heard on the news about drugs that, or vaccines that are in phase 1/2 trials, or going into phase 2/3. So we're combining these phases to both look at efficacy earlier and to introduce the vaccine to more people earlier. So there are vaccines starting with a couple hundred people rather than starting with 30, 40 people. We're also speeding up the manufacturing to basically build factories now, and we don't know yet which vaccine we're going with, but we're getting all the manufacturing ducks in a row, so that way when we have a vaccine or likely multiple vaccines that can go to market, we can get that manufacturing pipeline is already set up, and we can produce it really quickly and get it distributed out to the population. And the current goal for doing this is early next year, which is crazy fast. And just to annotate this a little bit further, part of how we're moving so quickly is doing this early bit, as I mentioned, times over 100, because a lot of the vaccines, as we saw at the clinical trials, are probably going to fail. So there are 160 currently in development with the hope of getting a few good ones out at the end. And there are also movements to build multiple different types of manufacturing systems for the different types of vaccines. So that way, regardless of whether an attenuated live vaccine or an mRNA vaccine, which we're going to talk about in a second, end up winning the vaccine race, we have a manufacturing system set up to make those vaccines. And also to make the more generic things like vials, stoppers, syringes, set up supply chains. So that way, once we get all this going and we have a vaccine, we can get it out to people. And how do we actually prove effectiveness when we're talking about a vaccine? And this is a lot of conversations going on right now, is for these trials, finding people to vaccinate who work in higher risk jobs. So we're talking about things like frontline medical workers who are more likely to come into contact with the virus, because giving the vaccine to someone who never leaves their house and never sees anyone is not going to be a really good test of how well the vaccine works. We can also vaccinate people who live in higher risk areas, so areas where there's still a lot of coronavirus in the population, and also just vaccinating a lot of people so that we have a larger sample size to work off of.
Yeah, and I'll chime in here. There's a semantic twist here, efficacy versus effectiveness. And can you talk about that, Kelly? Because it's a real kind of like head turner.
I'll leave the semantics to you.
Okay, the efficacy is under ideal circumstances. So when you're doing a test in the laboratory or under these ideal situations, you get the efficacy of the drug, or in this case, a vaccine. But then when it goes out to the real world, is the effectiveness then tested? And so when Kelly gets to the point when she starts talking about, okay, when it gets out there in phase 3 and it's trying to mimic what's going on in the real world, it's really about effectiveness and what's going to really happen under the real challenging situations of, okay, I give Kelly the vaccine and I give Allison the placebo, and or give 10,000 Allisons the placebo and 10,000 Kellys the actual vaccine. They have to go out into the real world and get exposed to the virus under normal circumstances.
Yeah.
And that's effectiveness, and that takes a long time to figure out. And there's no real way to rush that I know of.
Yeah. Well, the way that people are thinking about rushing this process is actually purposely exposing people to SARS-CoV-2 after vaccination. This is ethically really sticky because in clinical trials, you want to minimize harm to the people who are your human subjects. That's part of being a human subject is you should be expecting that you will not be harmed beyond what you may have signed up for. And exposing people to a virus that we're still learning about is really sticky. And this is made even more difficult by the fact that we don't have a good, consistent treatment. There's not a, well, if you get this type of response to the coronavirus, if you end up on a ventilator, if we do this and this, most people are back to normal. Since we don't have that type of treatment system set up yet, it gets a lot more difficult to basically design this type of experiment where you're risking the chance of someone getting the coronavirus, getting COVID-19, and having a really adverse reaction, possibly dying.
Yeah, and that, so Kelly gives, in our students that work in our lab, Kelly gives this long, really very in-depth training on ethics. And she would not like this if
No.
she was talking about this with students. And she would say, if somebody said, well, Dr. Salmon, what about purposely infecting somebody and to see if a vaccination would work? And I would bet that 6 months ago, Kelly would have said, absolutely not.
And to be honest, the first time I heard it talked about in the news, I was like, that's just like one wacky opinion. And then it kept coming up, and it's still in discussion as a potential way, because it can really shorten that time that if you're just vaccinating people and letting them go out and do their business, which for a lot of people right now is doing the minimal amount of business that you can do outside your house, it's going to take a long time to get an accurate effectiveness level for how well your vaccine is doing.
Yeah, and what's worse is, like in SARS-1, they canned the idea of trying to develop a vaccine because it died out so much. They didn't even have enough people to even test it in a place where they were going to be challenged. So so you wouldn't give these vaccines to people in New Hampshire because there's not enough challenge in New Hampshire, but down in Florida, that'd be the place to test it.
Yeah, I did see a quote from a group in Britain talking about they might move their phase 3 trials to the US.
Yeah.
Case numbers.
Yeah.
And this does, if we were to purposely infect people with SARS-CoV-2 after they're vaccinated, you do need a smaller cohort. An important caveat also for this is that because of the whole not having a consistent treatment, you would want that cohort to be young, healthy individuals. So people in their 20s and 30s, no pre-existing conditions, nothing to indicate that they would have a really bad case of COVID-19. But is that actually going to be representative of how that vaccine is going to function out in the world if you give it to children through the elderly, if you can only test it that way on a small cohort of healthy young people?
Yeah, and that goes to the word effectiveness, because you're not really testing the effectiveness then. Because there are a number of problems. If you say to yourself, how do people get infected now? Well, some people get infected, the minority, by touching things and then maybe putting their hands in their mouth or something. That's a minority situation. And the majority, and this seems to be backed up by the research, is that the majority inhale it, breathe it in some type of either closed or open system, so either outside or inside. And there's some people, and there's some evidence that's showing that if you get a bad case of it, it might be that you were exposed to a much more, I'd say, higher dose of it, and you might have breathed it in deeper into your lungs. So all of these variables, how are you going to test that under a purposely infecting this is why the effectiveness cannot, in my opinion, cannot truly be tested unless you go into the old-fashioned system. And this takes 3, 4, 5 months.
Yeah. So we are looking at a couple months for real good feedback on how well does this vaccine actually work. And that did remind me also that typically in phase 2 and phase 3, you also start introducing people in different age groups. So phase 1 is often still your pretty young, healthy individuals. Phase 2, phase 3, you start getting children, elderly, people with other pre-existing conditions, to try to get a sense of what that vaccine is going to do in those different populations.
Yeah, and that's another real challenge with the the COVID-19 is, as we all know, that if you have comorbidities, if you or you have certain things like obesity or certain other diseases diabetes or high blood pressure, these are all raise your risk of having a serious case of COVID-19.
Yeah.
And how do you test those people?
Yeah. And those people
Oh,
are certainly not going to want to be purposely exposed to the virus.
yeah. So, and Allison, who's really good at math, if you had, let's say, 0.01% of the children in the United States could get a serious case of it, and you have 60 million children going to school, do the math. What is that? Out of 60,000 kids? So,
Whip out my calculator.
yeah. So
Your note's not that
0.01
about it.
times 60 million, I think that's 60,000. So I might be off a zero, but I think it's like 60,000. There's a lot of children put at risk.
Yeah.
So and that's a big challenge with you talking about vaccines. If you have just a small number of people that are getting seriously affected by the side effects of that vaccine, when it gets into 300 million people, and you're going to start talking about 5,000 people having a problem, you got a problem on your hands.
Yeah.
Because see, many of those people would get the vaccine and might not even have gotten sick. So now you're saying to yourself, that that saying of medicine is first do no harm. You're in a sticky position, and that's why safety and vaccination programs is the ultimate goal. Is always make sure it's safe.
Yeah.
And I will put a plug in for vaccination for people
Yes.
who don't believe in vaccination. And not that at a certain point you wonder, do kids need all these vaccines? But at any rate, we do know that children and actually humans that are vaccinated versus people that are not vaccinated have actually a stronger immune system. That has been borne out by the facts. And this is decades of research. So just throwing that out there for those people that don't believe in vaccines.
Vaccines are good.
Yeah, vaccines are good.
Get vaccines. Okay, so going back to our many horses in the race, let's talk about some of them specifically. So there are some, they're making their way through clinical trials right now that follow the more normal approach to vaccinations that we're familiar with. So these are things like the replicating live attenuated virus, inactivated dead virus, or using purified pieces of the virus. And whoops. Okay, sorry. And the downside of these is that it takes time to make and verify these vaccines. So if you're making a killed virus, did you actually kill the virus? That's really important to verify before you give people a vaccine. And also it requires growing up a lot of this virus in a lab, which people are not super thrilled to do right now. So, but there are some companies that already have these approaches in development. And I just have a few examples here. And there's whole virus vaccines that are currently in phase 3 from this Wuhan Institute, which is actually a company called Sinopharm that has a phase 3 trial currently going on in the United Arab Emirates, and Sinovac, which has a phase 3 trial in Brazil. And there's purified antigen vaccines. They're in phase 2 in China from the ZFSW group, and in combined phase 1, phase 2 trial in the US, which is Novavax. And Novavax actually on July 6th was given a lot of money, $1.6 billion from the US government to promote their current manufacturing and development of their vaccine. But there's also a lot of new approaches that are strong competitors in being the final vaccine or one of the final vaccines at the end of this. And these are RNA, DNA, and recombinant virus vaccines. And it's 3 different types, but they're all kind of getting at the same thing, which is giving some cells in you the ability to make proteins that look like these viral proteins or are these viral proteins, and basically making your cells part of making the vaccine. So all proteins, including these antigens like spike, are coded for by genes that are in RNA or DNA. So for the SARS-CoV-2 virus, it's an RNA-based genome. And that RNA can be used to make the spike protein in human cells, which is what the spike protein is going to do normally, or what the SARS-CoV-2 virus, is going to do normally when it takes over some cells, is force your cells to make proteins for it, so make spike proteins. And these can be introduced either individually as DNA or RNA, which we'll talk about the mRNA Moderna vaccine, or they can be introduced with a virus, so having a different type of virus carrying that RNA or DNA as a way for it to get into your cells, to make your cells start making the spike protein. And this actually has an extra bonus of you have a virus in you that your immune system is going to be like, ooh, a virus, that's bad. And it kind of acts as an adjuvant in the making of that spike protein, is that you have a virus going on and you have spike protein. So your immune system is going to really respond well to that. And we'll talk about the Oxford vaccine after the Moderna vaccine, and that's using this other virus-based approach. And that's a recombinant virus, recombinant meaning it's a virus with genes from a different virus in it. So for the Moderna vaccine, it's mRNA based. So as I mentioned, RNA has genes that code for the proteins like spike. mRNA is just messenger RNA. It's the in-between between the gene, say the RNA genome in the SARS-CoV-2 virus, and actually making a protein. There's a little photocopy in the middle that allows the spike protein to get made, and that's the mRNA. In the Moderna vaccine, it's actually just the mRNA wrapped in a membrane, basically membrane bubble. And this membrane bubble looks like, or acts like your cellular membranes, and can actually fuse with the cells in your body to deposit the mRNA into some of your cells. So this is showing it being injected near your lymph nodes into a muscle. And it's showing here that it goes into the muscle cell. This mRNA goes through the ribosome, which is what allows it to be translated or turned into a protein. And then this cell is going to start kind of spitting out viral antigens, spitting out some spike proteins, which circulating immune cells can then pick up on. And it also goes through this process where your cells kind of present little things, little samples of what's going on inside of them. And some of these are also going to get picked up that way and presented to immune cells. And normally this is just signaling to the immune cells, I'm a normal cell with normal things in me, nothing to worry about. But it's a way of showing when that cell has become infected. So this makes this muscle cell seem like a cell that has been infected by a virus. So that way it prompts immune responses from antibodies and B cells, like we talked about last week, but also T cells, both killer and helpers, that are other parts of the adaptive immune system that help recognize infected cells and battle it as part of the immune system. And also, if this gets into a lymph node, you can get a similar type of translation and presentation of the spike protein to immune cells. And just as a clarification here, because their picture is kind of sciencey, antigen presenting cells in the lymph node include things such as B cells and macrophages, which we talked a bit about last week, and also other fancy cells called dendritic cells. And the antigen presenting is just that they help present different things to immune cells as possible targets. So this was actually the first one to phase 1 clinical trials in the US and made a lot of news when it did. That was March 16th. And that was less than 3 months after the or I think about 3 months after the genome was sequenced for SARS-CoV-2. So basically we learned all the genetics of SARS-CoV-2, and Moderna already had this platform set up for Zika virus and cytomegalovirus, and a few other vaccines that they had in development as using this new mRNA-based platform for a vaccine. And since all they need was a genetic piece of what makes the SARS-CoV-2 virus unique, they were able to get that really quickly from the genome sequencing and able to start pumping out some vaccine samples. And so on March 16th, the first person was injected in Seattle as part of the phase 1 clinical trial. And as sort of implied by that really quick turnaround time, this is a fast production line, and it can be very easy to scale up, which makes it a very attractive target as we're thinking about vaccines that we want to be able to quickly produce and get on the market once they're approved. And this is why I mentioned before that they already had a few in clinical trials. And right now, actually yesterday, oh no, this was 2 weeks ago. The other paper was published yesterday. 2 weeks ago, they released a preliminary report of the phase 1 trial. And they found neutralizing antibodies, so the ones that are able to bind spike protein, in all the patients that they vaccinated. And they also found that it activated the T cells, so that other component of the adaptive immune system. So it seemed to give a good immune response in all the individuals. They tested 3 different concentrations or amounts of their vaccine doses. And they're going to be proceeding to phase 2 with the middle dose, which was 100 micrograms. And phase 2 is already underway, and phase 3 is set to begin next week. And they're going to be recruiting 30,000 people for that.
Kelly, do you know in the phase 3 if they're going to have anybody over their 50s, because they didn't in phase 2?
I saw something that implied it, but I don't know that I saw it for sure. That was an important caveat, that despite what I said before about phase 2 often tries to get children, the elderly, I couldn't find if they had children. I'm feeling like no.
They did not.
Okay. And they didn't have anyone
sure
over
they did
55.
not. Yeah.
So the thing I read implied that there would be, because it kind of mentioned it as like a negative for the phase 2.
To me, that was a huge negative because I read they did not publish the real study. It's not like a published paper where you read the methods and materials.
Yeah, it's a report.
And so it was kind of difficult to interpret, but I think they did not go over the age of 55, and that was a real red flag to me, because I mean, that's anybody over 55, you have probably half the morbidity cases and certainly most of the mortality cases.
You don't trust us all to get vaccinated to protect you, Peter?
Yes, I'm hoping for that.
Yeah. Yes, definitely. So that's one newfangled type of vaccine that's on the market. And Moderna is not the only one that is doing an RNA-based vaccine. There are a couple other companies, and there are a couple of companies doing a DNA-based vaccine. So same idea, different genetic material that you're starting with, but the idea of getting that genetic material into your cells and turning your cells into little vaccine factories for you.
So Kelly, because this is really your world of mRNA and DNA, it's not mine. Mine's just more vaccines. Tell me, first of all, has there ever been an RNA vaccine that brought to market? And what do you see as some of the real technical issues they could run into with this baby?
So there have been none brought to market. And I think even the ones that Moderna had in clinical trials were, I think, only as far as phase 2. I might be wrong on that. Don't quote me on that. But it doesn't have as much of scary side effects from how I see it. So especially the RNA, because RNA is by default somewhat short-lived in your cells. Mm-hmm. So it gets in, it gets turned into proteins a few times, then it gets degraded. So I don't foresee that kind of getting out of control. Mm-hmm. DNA, if it can get into the nucleus, there's a really small chance that it can like stick itself in your genome,
Hmm.
and then you
How
might have
about,
one
how
cell that
about
like just makes
so
spike
that's maybe
for
the
fun.
bad side, but what do you think is the chances that it would actually work? I mean,
Oh,
if, yeah,
I think so. If they can actually get it to get into the cells and get the cells to make spike, which it seems like based on the clinical trials that they have, I think it's
okay,
a really great way of speeding up the vaccine production and getting your body to actually make a pretty good response.
okay,
So
good.
I think it's really promising. I think when I first learned that there are mRNA vaccines, I thought it was crazy, but I think it's actually a really cool way of making a vaccine.
Good. Ah, well.
You're skeptical. You're skeptical of the RNAs.
Well, no, yeah, it's just we'll put it this way. If I had to put my money on it, I'd go with the old proven systems
Yeah,
because I just know they work. We've been doing it for really thousands of years, but on a technical level with all the caveats and testing and everything, we've been doing this for 50 years and it's
yeah,
worked really well.
yeah. That's the weird thing about this is that the two leading contenders right now are
Hmm.
Moderna and the one I'm about to talk to next, Oxford, which are vaccine platforms that have not been brought to market before. So things that we're not familiar with, and we are going to, if they're successful, ramp them up and give them to a lot of people. So that is a potential drawback. So the one that typically gets referred to as the Oxford vaccine and is in partnership with AstraZeneca. It is a recombinant virus vaccine, which means that it's one virus carrying another virus's genomic material or part of another virus's genomic material. And it's using that virus as a vector or a way to deliver the SARS-CoV-2 genes into cells. So this is a schematic of how this vaccine was developed. They took an adenovirus, which is just a family of viruses from chimpanzees. In chimpanzees, it causes a common cold. And they've modified it, attenuated it to make it so that it's unable to cause disease and it doesn't replicate very well in humans. And then from SARS-CoV-2, they took that genetic material that we got really early on from gene sequencing and took the genetic material for the spike protein and inserted it into this chimpanzee adenovirus vector. So now you have your recombinant virus and a chimpanzee adenovirus carrying SARS-CoV-2 spike protein genes. And you infect your patients with this recombinant virus. And again, it's a way of getting the genetic material into human cells. So in this case, the adenovirus, it's not capable of causing illness in humans or replicating in humans, but it will still get into cells that it's near and deposit that genetic material. So those cells will still express the spike protein the same way as I showed with the Moderna vaccine. And it allows the body to produce antibodies and produce an immune response against the spike. With the hope being that if that individual is then infected with SARS-CoV-2, that they'll be protected. And this one was just published yesterday. The reports from their phase 1, phase 2 combined trial, which was really positive, and they are also moving into phase 3 next week. And again, getting about 30,000 people for the phase 3 clinical trial.
So Kelly, they call this is the University of Oxford, AstraZeneca. Is this going to be owned by Oxford University and not be like a drug company owned business, or what's the
That's a
story there?
good question. I did start trying to get into that and then I got distracted, I think. But most of the people that are being interviewed are from Oxford.
Okay.
So it seems like the leading scientists on the study are all at Oxford. And to be honest, I didn't know until I was putting this presentation together that AstraZeneca was even involved.
Ah.
So they don't seem to at least have a lot of clout from like the public standpoint. I don't know if they're behind it, like waiting for it to go to market to make some money off of it. But
Yeah.
As of right now, most of the stuff I've seen on it is coming out of the University of Oxford and not from AstraZeneca.
Yeah. Because I worry a little bit about companies owning one of the few cures for a plague.
Yeah. So that's a good segue, or part of this busy slide. And this is actually outdated numbers. But the main summary of all of this is that we are working on attacking the pandemic from a lot of different angles. So we're thinking about using convalescent antibody therapy that we talked about last week. There are efforts to repurpose drugs that are already on the market, like remdesivir, and we're trying to produce a vaccine. So we're attacking this for treatment and prevention from multiple angles, and specifically within vaccines. Part of having all those horses in the race is that we don't fully expect there to be one winner. So as I mentioned, in terms of production lines, we're probably going to, and just timing, it's probably going to need to be multiple different vaccines that end up coming to market and being used on the population. And we might find that some vaccines are better for different age groups, like maybe one group.
Yeah, that's a good point.
Yeah, it's because there, even for like flu vaccines and stuff, there are types of flu vaccine that work better for the elderly, types of flu vaccine that are better for children. That might be something that we come across with this as we learn more through the clinical trials. And another thing is cost and availability in different countries. That countries are trying to put stakes in who gets how much of supplies that are made in their country. And we all know cost is a problem with all healthcare related things. So a lot of things are going to feed into what vaccines are available when and to who. So that's part of why having so many vaccines currently in trials is going to be really important, and hoping that multiple of them are able to be good contenders and be able to be used on the general population.
So like if Oxford was the benevolent people they could license this free to anybody, and companies like AstraZeneca, if they wanted to be benevolent, could manufacture it at cost.
Yeah, that would be ideal. And I mean, some countries are trying to force for this to happen, but because vaccines are currently being developed by multiple different countries and multiple different platforms, it's going to be really interesting to see how this actually all comes together.
Yeah.
So that, on that semi-positive note,
Yeah.
that's what I've got to say about vaccines. I'd like to thank the Claremont Makerspace for giving us this platform and collaborating with us. As I mentioned at the beginning, I'm with the New Hampshire Academy of Science. We do a lot of work with middle and high school students to do authentic scientific research, and we do outreach things like this. And if you want to learn more about us, our website is www.nhacadSci.org, and you can reach us at that same name, nhacadsci@gmail.com. And we're also partnered with the Fairbanks Museum and Planetarium STEM Lab. And I will open it up if there are any questions.